An infection, by definition, is an instance in which a pathogen begins to reproduce inside of a host. For viruses and their human hosts, that means viruses need to get into the host and find the right cells or tissue. Once there, the virus has to gain entry into the host cells and reprogram those cells to make more virus. Then, viral progeny, that is, the offspring, must be able to leave the infected cell and go on to infect new cells. In this video, we will begin to look at how viral infection works by using influenza as a model. If you are interested in how the human body responds to viral infection and where uncomfortable symptoms like fever, fatigue, sore throat, and muscle aches come from, consider completing the An Inside Look at the Flu homework in your Chapter 13 folder. By the end of this video, you should be able to name 3 factors that determine whether a virus can infect a cell. You should also be able to describe the steps of the virus life cycle. And we're going to start talking about influenza. With respect to influenza, you should be able to describe the 4 types of influenza viruses. You should be able to name the spike proteins and receptors for influenza and explain their functions during an infection. And then you should be able to describe the structure of influenza viruses. As you already know, we live in a world that is full of viruses. You can't even breathe, eat, or drink without them entering your body throughout the day. The question is, if we live in a virtual cloud of viruses, why aren't we sick more often? The truth is that many of the viruses that come into our bodies are unable to infect us because we don't have the right types of cells for them to bind, enter, and grow. So they pass through us and we're unaffected. Occasionally, a virus will enter our body and it'll find the right kind of cell. It'll enter and start to grow inside of us, but we don't suffer any harm from it. These types of infections are called inapparent or asymptomatic because they don't produce any visible effects or signs or symptoms of infection. Then, the times that we're most aware of, viruses do enter our body, and they either directly or indirectly produce damage that can be recognized as a sign. The signs are things that are observable or measurable. So fevers, high blood pressure, rashes, inflammation, things like that. Or the damage will produce symptoms, things that can't be seen, like a sore throat maybe, or achy joints, fatigue. It's what you feel, not what can be seen or measured. There are a number of conditions that have to be met in order for a virus to initiate an infection. First and foremost, a potential host must come in contact with enough viruses for them to cause an infection. Labs describe the average number of particles necessary to initiate an infection as the infectious dose. This number varies with the virus. The infectious dose for influenza viruses suggests that somewhere between 600 to 800 virus particles have to enter a host to cause an infection. For respiratory syncytial virus, more than 160 to 640 particles are needed. Some viruses, like norovirus, are extremely infectious, requiring exposure to only about 18 virus particles to cause an infection. Now, these numbers are hard and fast, but they are the result of controlled conditions in a laboratory. In real life, other factors such as the immune status of the next host, environmental conditions like temperature and humidity, and fomites may also play important roles. We come in contact with these virus particles when we inhale mucus particles that someone produces as they speak with us, or on globs of mucus left behind when someone blows their nose and then touches a surface like a cell phone or a desktop. Sometimes we ingest them when we use the same utensils as someone who was ill, or as happened to my husband a few months ago, we hold the bucket as a sick child vomits into it. It happens a number of ways. As future healthcare workers, you could become exposed through accidental needle sticks while giving someone an immunization or helping them with an IV. In addition to requiring the proper number of particles to start an infection, viruses must also make contact with the right cells and tissues in the body. Remember that viruses can only infect living cells? Well, this means that most surfaces in the human body are well protected against infection. Why? Well, the body's protected by a dry, tough barrier called the skin. The superficial layers of the epidermis are composed of multiple layers of dead keratinocytes that cannot be infected by viruses, provided that the skin remains unbroken. Some viruses, like human papillomavirus or herpes viruses, can gain entry to the body through abrasions or cuts. A smaller proportion of the surface of the human body is covered by mucous membranes. Mucous membranes line the respiratory tract, the gastrointestinal tract, the genitourinary tract, and they surround the structures of the eye. Let's take a few minutes to look at the respiratory epithelium, because it is the mucous membrane that is the most common portal of entry for viruses entering the human body. Portals of entry are sites that pathogens use to enter a susceptible host. Now, that said, some pathogens have what's called a preferred portal of entry that deposits them near the cells and tissues that they infect. If they try to enter the body through some other portal, they won't cause an infection. Now, back to the respiratory epithelium. As you can see from this diagram, the respiratory epithelium is made up of a single layer of ciliated epithelial cells supported by a basement membrane. The basement membrane is on the far right of your slide here. I won't waste your time by trying to color it completely, but hopefully you get the point or the idea of where it is. The apical surface of the epithelial cells is covered by a thick layer of sticky mucus. The mucus is secreted by goblet cells that are interspersed among the epithelial cells of this mucous membrane. The mucus helps to trap debris and virus particles that are inhaled into the body. Antibodies associated with the mucus can inactivate viruses at this point. Another factor that may prevent infection is something called the ciliary escalator. So the ciliated cells that are located in the bronchi, the bronchioles, and nose beat their cilia. And as the cilia beat, it pushes the mucus and anything suspended in it, such as debris or viruses or other microbes, up and out of the throat. Oops. Up towards the throat and out. At this point, the mucus can either be swallowed or it gets coughed out. Now, with all of these preventative factors, occasionally some viruses actually do make it past the mucus down to the epithelial cells that they can infect. I should note that this can happen in the upper respiratory tract here, as well as in the lower respiratory tract down here. What should be clear here is that the more virus particles that enter the body, the more likely it is that a virus can establish an infection. It's more likely that the virus will make it past the mucus down to the epithelial cells. Most viruses are only able to grow in specific tissue types or specific cells in organisms. We call the number and the types of cells that a virus can infect its host range. And typically the host range for a virus is very narrow. If you think about HIV, out of all of the different types of cells in the body, it's only able to infect one type. And we'll talk about that in another lecture. Host range is determined by interactions between viral glycoproteins and viral proteins on the surface of the virus, and proteins or carbohydrates, which are called receptors, you can see them here, on the surface of the host. You can think of the viral part of this interaction as being like the key, and the host part of this interaction as being like a lock. In order for a virus to enter a cell and infect it, it's got to have the right key. Now, many viruses actually require two keys to start an infection. The first is called an adhesion receptor. The virus uses this to actually attach to the cell, to the cell's membrane. And for these types of viruses, just the binding of the virus to the adhesion receptor brings the virus close enough to bind a second receptor called the entry receptor. And it's the entry receptor that will actually trigger the cell to bring the virus inside, either via endocytosis or fusion, or in some cases, macropinocytosis. In order for any of this to work, the receptor that a virus uses has to be available for binding, it has to be functioning, and the host cell has to be permissible. Permissible means that it can be infected, and if infected, it'll produce more viruses. When a virus binds its receptor on a host cell, it initiates a potential infection. Most virus infections follow 6 basic steps, although there can be some modifications to this scheme. You are responsible for knowing the 6 steps of a virus infection, as well as describing what happens during each steps. In the first step, which is called attachment, the virus binds its adhesion receptor on the plasma membrane of a host cell. This event alone can trigger the next step, called penetration. In penetration, the virus particle is brought into the host cell by endocytosis, fusion, or a process called macropinocytosis. Macropinocytosis is a phenomenon where a cell drinks in some of its environment using vesicles. And if that environment contains viruses, the virus gets into the cell. I've put a link to a video on endocytosis and fusion in your lecture videos folder for chapter 13. Now would be a good time to take a break from this video and view that one. Come back when you're done. Penetration is followed by uncoating. The part of the virus that's essential for infection is its nucleic acid. But when the virus enters a cell, its nucleic acids are usually packaged in a capsid that has to be removed. Virus capsids are removed by acidification, which denatures the proteins, or through the activity of enzymes that digest the viral capsimeres, the proteins. Once the viral nucleic acid is released, biosynthesis begins. In this step, the virus reprograms its host to produce viral nucleic acids, including messenger RNAs, which can then be translated by cellular ribosomes to make viral proteins. Once the cytoplasm has enough viral nucleic acids and proteins, they spontaneously assemble to form new virus particles in a step called maturation. The new viral progeny are eventually released either by budding from the host cell, as is what happens with your enveloped viruses, or by rupturing the host cell, spilling progeny viruses into the environment where they may go on to infect other cells. So that was a lot of information. Let's do a little check to see what you remember. Here's your first question. A virus that infects a host without producing signs and symptoms of infection causes a, would that be a disease, an insurance claim, an inapparent infection, or a chronic infection? That would be an inapparent infection. Let's try another. Which of the following is not a requirement for a virus to cause an infection? Is it enough particles must enter a host? Or an available susceptible receptor? Or hand washing? Or a permissive host? Hand washing, of course. Hand washing is actually one of your best defenses against a viral infection. Which of the following is determined by interactions between viral glycoproteins and host proteins or carbohydrates? Would that be host range, type of virus life cycle, uncoating, or the shape of a virus capsid? Host range, absolutely. Finally, which of the following is mismatched? Would that be attachment, is when virus is attached to the cell membrane? Or is it penetration, when virus nucleic acids invade a host cytoplasm? Or is it uncoating, when the virus capsid is denatured or digested? Or how about biosynthesis, when the host cell is reprogrammed to make viral components? Maturation, viral nucleic acids and proteins assemble into viruses? Or release, viral progeny escape host cells by budding or rupture? What do you think? Penetration, right. Remember, penetration is when a virus enters a cell's cytoplasm by endocytosis, or maybe it's fusion or macropinocytosis. Now that we have a basic understanding of how a viral infection works, let's look at influenza as a model for virus infection. In this video, I'm going to give you an introduction to influenza. In my next video, we'll actually tackle the life cycle. Influenza, also called the flu, is a contagious respiratory infection caused by influenza viruses. There are 4 types of influenza viruses, but only 3 infect humans. Types A and B are responsible for seasonal flu epidemics every year. You've heard of flu season, right? Well, we could call that the annual flu epidemic, but flu season sounds more catchy and not quite as threatening. Influenza A and B viruses tend to circulate and make people sick in the winter. Flu season, as a result, generally goes from November of one year and extends to February or March of the next. Occasionally, in a particularly bad year, flu season may extend all the way until May. Type A influenza is the only influenza strain that is known to cause flu epidemics and pandemics. It is also associated with more severe complications than type B. When you hear a doctor or an expert refer to flu viruses with the letters H and N, for example, they say something like H1N1 or H5N3 or H7N2, that is a type A flu virus. Type B influenza usually causes milder symptoms than influenza A, but it can still cause significant harm to the body during an infection. Type B viruses are also not named with the letters H and N, but rather they have the location where the flu virus is thought to have originated associated with their name. Type C influenza causes very mild illness, and type D is only known to infect cattle. The signs and symptoms of influenza infection include headaches, fever, fatigue, achy joints, runny or stuffy nose, coughing, and it may include vomiting. This means that although the virus infects the epithelial cells of the respiratory system, its effects can extend to multiple body systems. Most people who get influenza will recover in a few days to less than 2 weeks. but some people will develop complications like pneumonia, pericarditis, croup, meningitis, or rhabdomyolysis. Rhabdomyolysis is a breakdown of the skeletal muscle. Some of these complications can be life-threatening or even fatal. By the way, I have put a link in the virus links folder of your course space that describes how respiratory viruses like influenza virus can cause pneumonia. And as we all know from watching the SARS COVID-19 pandemic unfold, this type of pneumonia can be fatal. Complications are more common in the elderly and young children. People who have other predisposing conditions may also be at risk. Everyone's best option for avoiding complications due to influenza virus is to avoid getting infected at all. Get your yearly flu shot. Try to prevent the effects of this virus. Even in those years where scientists make the wrong guess as to which influenza virus will be circulating during flu season, having a flu shot can help reduce the severity of your signs and symptoms and can help prevent the more serious complications that come from influenza infections. Influenza viruses are helical pleomorphic enveloped viruses. This means that they have a helical capsid, they can have multiple shapes, and they have a lipid layer. You may remember from the last video that some viruses acquire an envelope by snatching a bit of plasma membrane from a host as they bud away. Embedded within this envelope are two glycoproteins that form spikes, like you see here, on the virus surface. Hemagglutinin is the most abundant of the spike proteins. So this is hemagglutinin here. It makes up 80% of the spikes. Hemagglutinins' job is to attach to a carbohydrate called sialic acid found on the cells of the respiratory epithelium. The second spike protein is called neuraminidase. Neuraminidase cuts sialic acid off of infected cells. We'll address why in the next video. Hemagglutinin and neuraminidase are antigens and are sometimes called HA and NA. When influenza A viruses are given names like H1N1 and H2N3, these numbers refer to which subtype of hemagglutinin and neuraminidase the virus has. These two proteins are also why you need to get a new flu shot every year. Now would be a good time to watch the video Influenza Get the Genetic Drift from your lecture videos folder. Pause this video and watch that one. Then come back when you're done. The third type of protein that's embedded in the viral envelope is an ion channel protein. These help with virus uncoating after the influenza virus has infected a cell. Influenza viruses have a helical capsid, and internal to that, there are 8 strands of single-stranded RNA plus bound proteins. We will call these 8 strands and their bound proteins the influenza nucleoprotein. The receptor for hemagglutinin is a carbohydrate displayed on epithelial cells called sialic acid. So the carbohydrate is sialic acid. This animation shows a hemagglutinin spike protein from influenza. The protein is the large elongated structure that looks a little bit like the end of a scepter. The tiny red and green molecules on the very end of this spike protein, they're up in this region, those are the binding sites for sialic acid. Tiny, aren't they? As it turns out, sialic acid has a lot of different forms, and we're going to see that the different epithelial cells throughout the respiratory mucosa display those different versions of the sugar in different regions. This means that our influenza viruses are going to stick to the respiratory mucosa at different points in the respiratory system. Type B influenza, which I'm calling the human flu because it's only found in humans, is a very common infection, and it's responsible for some of your more severe head colds. It's interesting because type B influenza tends to prefer the sialic acids that are bound to epithelial cells in the nasopharynx, the trachea, and the bronchi. You'll also find cells in the paranasal sinuses expressing this type of sialic acid. Perhaps the ability of the human flu to bind cells in the upper respiratory tract are responsible for the symptoms that we see, like sore throat, cough, runny nose, sneezing, fatigue. Avian or bird flu is an infection of domesticated and wild birds that occasionally is transmitted to human beings because of our close contact with infected birds. These types of viruses are type A influenza viruses, and they prefer the sialic acids on the epithelial cells that are deep within our respiratory system. So they prefer the sialic acids on the alveoli and the bronchioles, so out in here. This may be why your avian flus are more associated with the more severe complications like bronchitis and pneumonia. Swine flu is an influenza infection found in pigs primarily, but it can spill over into the human world when we work with infected pigs. Since this virus binds the same receptors as both the human type B and the avian flus, it's associated with some of the most severe illnesses and complications. A relative of swine flu was responsible for the influenza pandemic of 1918. By the way, diseases that start in animals but spread to humans, they're called zoonoses. Zoonoses. That was a lot of information. Let's check in and see what you remember. Which of the following is the influenza virus's spike protein that binds with receptors on host epithelial cells? Is that hemagglutinin, neuraminidase, ion channel, or sialic acid? Hemagglutinin, of course. Hemagglutinin is the spike protein, and it gets its name from its ability to make red blood cells stick together, or agglutinate. Which of the following is the receptor that influenza binds? Would that be hemagglutinin, neuraminidase, ion channel, or sialic acid? Sialic acid. I'm glad you didn't choose neuraminidase. Remember, neuraminidase is the enzyme that cleaves the sialic acid off of epithelial cells after they've been infected by influenza. Which types of influenza virus infect humans? Would that be type A, type B, type C, or type D? There's more than one answer to this question. Types A, B, and C are the correct answers here. Remember, type A includes your bird flu and your swine flu. Type B, only found in humans. And type C, well, it's responsible for causing mild symptoms. Last one. Which of the following is mismatched? Is it zoonoses, which might mean animal viruses that can infect humans? Or is it infectious dose, which might mean that is the average number of particles necessary for infection, or is it fomite, which could mean inanimate object involved in infectious disease transmission, or is it portal of entry, which might mean the site that pathogens use to enter the human body, or is it most common portal of entry, being the genitourinary tract? What do you think? Of course, it's the last one. I knew you'd remember that the most common portal of entry for microorganisms into the human body is the respiratory tract. So that's it. In this video, I named 3 factors that determine whether a virus can infect a cell. We said that it was number 1, the number of viral particles. You have to have enough of them to actually make it past the host's defenses in order to reach their target and it cause an infection. Number 2, the tissues and cells inside the host have to have the appropriate receptor. That receptor has to be available for binding and susceptible to binding, meaning the host has to have kind of a low immune system, lower defenses. The third thing was that the cell that the virus is going to infect has to be permissible. It has to have all the materials inside of it that will allow the virus to reproduce and produce progeny or offspring. I also described the steps of a virus life cycle. Remember, they were attachment, penetration, uncoating, biosynthesis, maturation, and release. You, again, will need to know that for your exam. I also talked specifically about influenza. I described the 4 types of influenza viruses. I talked about the proteins that are involved in getting influenza into a cell for infection. We said that hemagglutinin is the spike protein on the surface of the influenza virus, that it's the key that unlocks the epithelial cell during an infection. We said that the receptor on the host cell is sialic acid, and depending upon where you are in the respiratory system, that sialic acid may be different, and it can bind different influenza viruses as a result of that. We also talked a little bit about neuraminidase, which is another spike protein. Remember, neuraminidase job is to cleave off sialic acid after a cell gets infected. Then I described the structure of influenza viruses. We said that these are helical viruses. They have a genome that consists of 8 segments of RNA. In my next video, I'll tell you that those are antisense segments, and we'll talk about what that means. We said that these 8 segments have proteins bound to them that are involved in replicating the virus, and that there's a capsid that surrounds those nucleic acids. Remember that the capsid is helical in nature. Outside of that, you had an envelope, and that's where we're going to find the hemagglutinin, the neuraminidase, and there'll be some ion channels there as well. So, I hope this will be helpful to you. You'll really want to spend time with this video and my last video because they give a lot of introductory information that will be useful for you in chapter 13, but when we get to chapters 14 and 15, you'll hear some of these concepts again. In my next video, I'll talk in detail about the infectious cycle for influenza virus. I'll see you there.